The overall goal of this procedure is to generate human-induced pluripotent stem cells using hair-derived keratinocytes. This is accomplished by first isolating hairs from a donor. The second step is to plate down the hairs in order to facilitate keratinocyte outgrowth. Next, the keratinocytes are maintained and expanded in vitro. The final step is to reprogram the keratinocytes into human-induced pluripotent stem cells using an episomal reprogramming method. Ultimately, human iPS cells can be isolated and characterized using immunohistochemistry to show the expression of pluripotent markers.
The overall advantage of this method over existing methods such as retroviral or lentiviral method is that by using episomal factor, we're avoiding undesirable transgene that integrate into the genome in the stem cells that are generated.
Begin by thawing a vial of extracellular matrix solution on ice overnight. The next morning, use pre-chilled pipette tips to add 200 microliters of the solution to 12 milliliters of chilled DMEM F12 medium. Add 1 milliliter of the diluted extracellular matrix solution to each well of a 12-well plate and incubate the plate overnight at 37 degrees Celsius to coat the wells with matrix. Next, collect primary human hair from the donor's head by placing fingers close to the root of the hair and plucking the hair in one quick and smooth motion. Check to confirm that each collected hair contains an intact hair follicle. Place the follicle under a dissecting microscope and determine the growth phase of each hair. Collect 5 to 10 antigen hairs from each individual to extract keratinocytes.